This is one of the most experiments for the entire AS. Even if you're taking A2 in the future, that's the one they ask the most. And the reason is because it got a lot of little details. So when they have a lot of details, then they ask you many questions about it. So just pay attention. This is how you prepare microscope slides like this one here. So usually in schools, you can make microscope slides to watch cells. This experiment here is the one where you look at chromosomes. So there is a little technique here that we're gonna do to do this experiment. In step one, you will need the plants to observe those chromosomes. So the best choice is either onion or garlic. Why onion or garlic? Because those plants can grow roots very fast. So basically, if you can try this yourself, you like, take onion or garlic and you put them in water, they'll grow roots very rapidly. And we really need those rapidly growing roots because you can see the cells dividing. When a tissue, when an organ is growing, it will have many cells dividing. So that's the whole purpose of onion garlic. Look what he's gonna do. We're gonna cut 5 millimeters from the tip. Again, 5 millimeters is the standard. Now, questions may ask you to cut some other distances, but again, if you're writing this experiment, you just say you're gonna write 5 mm from the tip. Why is it that the 5 is the best? Because this is the region here, we call this the growth region, not the tip itself. No, right here, you have the majority of the cells that are divided. And those are the cells we're interested in. We are interested in the cells that divide fast. Now that I have my sample, we have one issue. The issue is that the sample is plant cells. Plant cells always come with one problem, and the problem is that they come with cell walls, tough cell walls full of cellulose, pectin, all of these things. They're not gonna let any pigment, not gonna let any color get inside the cell. And again, I can't watch these chromosomes unless I have stained the DNA. So for that reason, I will need to treat this root with hydrochloric acid just to soften the plant tissues, just because I don't want it to be tough. I want to break through those cell walls so that the color will penetrate. Okay, so step one, we cut. Step two, we added the acid. Step three, we're gonna now warm up that acid. So I'm gonna take my root plus the acid. I'm gonna gently warm it up. Again, that will contribute to the softening of those plant roots. Okay, now just warm it up gently. And next, we will take my root, now it's nice and soft, and I'm gonna add my pigment. Now, the pigment of choice we're gonna use here is this one. We call this Schiff's reagent. Okay, it's called Schiff, like this. And it's specific for DNA. So the reason why we have to remember this particular name is because this is one of the few pigments that you can use to stain. Stain means to color the chromosomes and DNA. So you add few drops of that color here. You'll notice that your root starts to get that color, but still you need to warm up the root plus that pigment. Why is this very, very necessary? Because the plants are full of water. The pigment will dilute. The pigment will become less intense if you just put it as it is. So we usually gently warm up the pigment and the root so that will intensify, that will improve the contrast of your chromosomes. So now it's nicely colored, stained. What am I going to do next? I'm going to start to spread those plant tissues apart. It's very, very necessary to make sure that you're watching one layer of cells. If you're looking at the plant cells from above, you'll be watching like stacks and stacks of cells. It's very, very difficult to see individual cells or see one layer of cells. So you literally have to do it yourself. You spread them apart like this with a needle, and then you will nicely have spread plant cells. Still not enough because we still have to do one more thing. So I'm going to take it now. I'm going to place the cover slip on top of my root. And now with the back of my pencil, I'll press on it. I'll squash gently. You're not going to like do like this, you break it. No, gently press on it like this. And that will do what? That will spread those cells far more. And now it's ready. You're gonna grab your microscope and you're gonna look at the cells. They'll be visible in that form. But you're not gonna see this first because you're gonna always start at low power. Why low power? Because just like if you're doing with any map, you look from above and then you zoom in. So you start with low power and then you increase the magnifications gradually until you get the best view. Okay? So let me review this one more time. What we've done here, step by step, total of 6 steps. First of all, we cut 5 millimeters. Then we softened with the acid. 3, we stained with Schiff's reagent. Number 4, we intensified with the Bunsenberg gentle heating. Then tease with a needle. Finally, you look at low power first, and you will see one layer of cells. Before this, you just need to press gently so you can spread the cells apart. Any question here? This is called the root tip squash experiment. Okay, so quick question. Tell me, what's your question?
Do we heat the Schiff's reagent with the cell, with the stem, or do we heat them separately?
So you want to heat the stain alone and the root alone, or no, you must heat them at the same time.
Okay.
All right. Okay, so let's try this. Let's review what we have done. I'll move tiny bit this way. Tell me, first of all, we're gonna cut away from the root tip. We will soften by adding. We will add this stain, warm up gently, place on, and then using a needle, then we'll place and press gently, examine using a microscope with this power first. So anyone can do any of these steps. So what did we do? How far we cut first? All right, go ahead.
At the root tip, 5 millimeters.
So 5 here, good job. We softened by adding, so what chemical did
Hydrochloric
you?
acid.
Right, good job. Then we added
Schiff's
this.
reagent.
Yep. Warm up gently too.
Soften.
No, soften is the acid. We've intensified, like we improved the contrast. Okay. Place on Then I'm going to
Micro
place on
slide.
a microscope slide. Okay. And then we,
Teeth.
very good, place, what
Cover
did I
slip.
place? I placed that cover slip. We examine using a microscope first with,
Low power.
yep, very good, thank you. So this was the root tip squash. Now I want to show you a calculation related directly to this topic. So what is this about? This is how do you measure the rate of mitosis? In other words, how can you tell if this process is happening fast or slow? You may ask me, like, you tell me what the point of this whole thing. Why is it necessary to find how fast mitosis is? Because this is a very good way to measure growth. You can measure how fast growth is happening by measuring the rate of mitosis. Now, here's a view of that root tip squash. And you can see here that there are cells doing mitosis, and there are cells that just still haven't done mitosis. Like these, whenever you see a nucleolus, like the one I have here, that's a cell that is still interphase, still haven't gone through mitosis. Like even this one, see? That's not mitosis. But when you see chromosomes, that's a cell doing mitosis. So this is called a mitotic index. Mitotic index, you check how many cells are doing mitosis here. So check the number of cells that are undergoing mitosis and then divide by the total number of cells. So basically, it's a ratio, it's a percentage, multiply by 100, and you would be able to tell how much of mitosis is happening. And again, this is very, very helpful because if you have very large number of cells doing mitosis, this means it's growing fast. And sometimes, or many times, growing too fast, dividing very often is not a good sign. So cancers is usually uncontrolled cell division. So if the cells are dividing very fast, then that's not a good. So you can always diagnose or determine if there is abnormal growth by checking this mitotic index. Okay? So let's try something. Let's try to check this question first. It says, put cross next to the stage of mitosis that is most likely to be observed. Can you tell me what do you think is the best option here? There's a little trick here, so just be careful. Do you know what the question means? It means like which stage of mitosis is the one is most likely to be found. You will always cancel this, right? Because that's not mitosis, right? So don't choose it. So then once I put this cross, they chose it. No, don't choose it, I meant. So why is this not? Because this is interphase, that's not mitosis. And from these, the stage that is most likely to be found is the one where I have the majority of cells. Again, if most of the cells are in one stage, this means that the stage is the longest, is the stage is most likely to happen. So for my case here, I'll go with metaphase. So metaphase because that's the largest one among all cells doing mitosis. Okay? Now I need your calculator because we are going to calculate that mitotic index here. And again, they will always include interphase, but that's not mitosis, okay? So how do I do that mitotic index? Very simple. Look, you add up all cells because I want that total here. and I want the cells doing mitosis. I'm going to call them mitotic cells, and I multiply by 100. Okay, so let's get that total. That total, if you add up all of these numbers, how much you get? Like add up all of these together. How much? So you're gonna get 795. Okay. Now, if I want only the mitotic cells, then I'm gonna only add these numbers. Okay. Can you add them up in your head, your mind? 35, 37, 6, and 24. How much you get? You're gonna get 102. I did not add it. I remember the number. Okay. Now you multiply by 100, you're gonna get that mitotic index. Okay, you may round it, no problem. So you write 13. Okay. Any question? I don't think it's difficult. Just have to be careful about this. Now, I have another question here. They gave us root. Okay, so we have like this plant root, and they said, look at the graph here, and try to figure out why is this a negative correlation? Was that, what do I have here? I have the root. They took cells along the root like this, and they calculated the index, the mitotic index. What did they find? They found that as they go further like this, the mitotic index is decreasing. Okay? So they said, why is this considered as a negative correlation? Simply because what is a negative correlation? A negative correlation is the one when you have one factor increasing, the other one will decrease. So I will just tell them the meaning of a negative correlation. I tell them because as the distance from the root tip is increasing, the mitotic index is decreasing. Now, the second part, I'm gonna leave it for you as a homework because it would need a ruler, but let me explain it to you. You have 3 cells in mitosis and 91 cells in interphase. So if you are to do the mitotic index here, what would be the total? If it's 3 cells in mitosis and 91 interphase, so the total is going to be 3 plus 91 is 94. So that's going to be a 94. The mitotic cells are going to be what? Those are going to be 3, right? So they said work out the mitotic index. So use the mitotic index formula and calculate how far from the root tip this was found. So which region in the root tip is this one? Again, once you calculate this index of mitosis, you're gonna plot that on the y-axis. So whatever value you get here, you plot it, and then you grab your ruler and you draw a line of best fit through these results and find it like this. So match it here on the distance. Okay? So try this and I'll check it for you. The remaining part, they said describe an investigation that could be carried out to confirm the results shown in this graph. So the graph is here behind me, and we want to plan an investigation to get these results. So when you get something like this, when you've been given data an experiment, you need somehow to adapt the experiment to serve the data. I mean, you should write the experiment as it is, but in a way to match the results of that experiment. So for that reason, I'm gonna write that same root tip squash thing, but this time I'm gonna adapt it for the numbers I was given here in this experiment. So I'll say using root taken from plant. So this is step here where I want to standardize the test. I want the test to be fair, okay? I'm gonna cut roots from the tip. Now, many people will write that 5 millimeters because they've memorized it, but here I really need to match the distance I was given here. So there's no 5 millimeter, right? So just pick any value, like 0.5 for instance, and then here at the end, repeat for the other values because that's what you're meant to do. So once again, I'm going to use root taken. I want this to be fair test. So all of my roots must be taken from that same plant. That's my first step. Cut the roots. How far we said? Not 5. Just take any value from the x-axis. Down here, I'll repeat at other distances. Again, the values I have right here at the bottom. Next, I'll place, that's the step where I have to soften. So I'd say warm acid. We're going to add drops of the stain. Do you guys still remember the name of the stain? What was stain is we use? We use that Schiff's reagent. Then it's necessary to warm up the slide and the stain together to intensify the color. Then to spread those tissues apart, we're gonna use a needle. Then I'll squash, I'll press, and then because this is all about mitotic index, I'll calculate the mitotic index. Okay? In most cases, whenever you have to study growth, to study how fast plants are growing to some conditions, it's always necessary to write this one here, the mitotic index. So anytime they ask you to study any stage or growth or cell division, that is necessary. Okay? Now, second thing we're gonna do now is the meiosis. Now, luckily, you don't need to go into so much depth about meiosis. You only need an overview about this process, not like mitosis where you have to study every single stage. To start with, why is this very important for all living things that reproduce sexually? So why meiosis is the thing that is done by all sexually reproducing organisms. It basically gives you two things, two very necessary things. Number one, it halves the number of chromosomes. So meiosis's role is to take chromosome number and cut in half. Why is this good? Because if this is the baby here and that baby was born to those parents, each parent will pass one of its pairs of chromosomes. And in other words, each parent here will pass 23 only. And that's all thanks to meiosis. So that baby will be inheriting the same number of chromosomes as his parents. So they got 46. Obviously, they're humans. He will be 46. Imagine life without meiosis. We would have had double the number of chromosomes every generation. It'd be impossible. So that's role number one. The second role, which is as important, is the fact that meiosis gives us we call genetic variation. Now, think of chromosomes in humans. You guys did this in Unit 1. Humans come with 46 chromosomes, or the best way to understand it here is that humans come with 23 pairs of chromosomes. Why chromosomes always come with pairs? Because each and every one of us inherited half of his chromosomes from his mom and the other half from his dad. So always your chromosomes come in pairs, mom and dad's chromosomes. These chromosomes are always in the same shape, same length. We call these chromosomes, so remember what we call them? We call these chromosomes homologous chromosomes. So when a child is born to his parents, he will inherit half from his mom, half from his dad. And again, the homologous chromosomes have the same genes. So the genes are exactly the same. The difference between each or every homologous chromosome pair is that they may or may not have the same alleles. So, for instance, here's a pair of homologous chromosomes, and you can see that this guy has inherited two different alleles of eye color. He has inherited two different alleles of hair type, blood groups. Okay, but these may be the same, may be different. And again, what determines how is he going to look like? It's based on dominance, which allele is dominant, which allele is recessive. So one more time, homologous chromosomes are pairs, same length, same genes. How are they different? They are only different because of the alleles they have. They may be the same, they might be different. In other words, every one of us here has 23 pairs of homologous chromosomes. Now, let's get to meiosis. For a cell to divide by meiosis, it has to go through the 4 stages of cell cycle. Every cell that goes in a cell cycle has first to grow, G1. So here we increase in size, get more cytoplasm, get more organelle. Just like a baby cell is growing here, okay? Then it goes into what we call the S phase. S phase is all about synthesis of one thing, that's DNA. So here we will duplicate the DNA. Okay? Now the DNA is replicated. Now the cell will grow further in G2. And here is where things start to become different than mitosis. So interphase is the same everywhere. But when a cell is to enter meiosis, it will have to divide not one time, it has to divide twice. So again, meiosis is not one division, it's two divisions. Because the outcome here is going to be 4 cells, 4 sex cells, 4 gametes. For that reason, the cell will be born, duplicate its DNA, it will divide once. So that's my first meiosis. We literally call it meiosis number 1. So that's when the cell divides the first time. And again, what do I call this? I'm going to call this meiosis 1. And then it will straight away divide second time. And this time we call that meiosis number 2. So each one of those cells we made here, each one of them will divide second time, and that is called meiosis number 2. Okay, so let's see how is this different from mitosis. In mitosis, we only had one division. Got it? So it was only one division. And in meiosis, we kept those homologous chromosomes. They were passed together. We were just splitting apart the chromatids. But no, in meiosis, we first separate those homologous chromosomes. So each one of them goes to one cell, and then you separate the chromatids. So you do separate the chromatids in the second stage. So that's the major difference between what we have here and what we had in mitosis last time. So one more time, let me just clarify this. So this is your cell. That's a cell was born with something like this. So assume that these are pair of homologous chromosomes. That's baby cell. The cell will go through G1. Here it will grow, get bigger in size. The cell will do the S phase. What's going to happen in S phase? Remember? S phase is where it's gonna duplicate. Nothing really happens in G2 except that the cell will increase further in size. Okay? So this is all together, what I call all three stages here. I call this interphase. Okay? Now, next, this same cell that was made in interphase will now enter what I call what's going to happen next. Now we're going to enter meiosis. And meiosis is two division. So this is M1. This is meiosis number one. What's going to happen here? In the first meiotic division, we will divide this into two cells. Each cell will have one of the homologous chromosomes, like this. Okay? So each one of those pair went to one of these cells. What about M2? M2 is like this. This divides and this divides. The first one will get, I'm just going to put sperms here because this could be sperms or egg cells based on the gender. Okay, so this will inherit one, two of these chromosomes. So basically, this here will divide into two separate chromosomes. Okay, and so is the other pair, so you get one copy each. Okay, any question?
Sir, what was the definition of homologous chromosomes? Could you
I
repeat?
showed you there. Did you want to see it again?
Yeah, I missed one word. Okay, thank you, sir.
All right, now let's go into a bit more depth about meiosis. Here I want, isn't M1 mitosis? We don't have we do have meiosis today. M1, we split the homologous chromosomes. M2, we split the chromatids. Now, look what I'm going to do here. I want to show you how does DNA, amount of DNA, say mass of DNA, changes during stages of meiosis. So here's my cell, the cell we started with. So that's a cell in G1. What's going to happen as S phase happens? So again, that's G1 here. S is like this. S is where you get the DNA duplicated. So we're going to get something like this. Okay? So the amount of DNA per cell has doubled. Now let's go to M1, meiosis 1. What's going to happen? You're going to get two cells. Each has one of the homologous chromosomes. Again, I'm simplifying this. I'm only showing you one pair of homologous chromosomes, but reality, there are 23 pairs. So here I'm going to get half, here I'm going to get half. Next, we're going to now go, so that was M1. Now is M2. What's going to happen? You're going to get the gametes, you're going to get the sex cells, and each will be haploid. So that's the secret here. We've halved the amount of DNA. Now be careful. Look at the y-axis. What did I put there? I put per cell. You may tell me like the amount of DNA here and here is more than that. No, I'm counting it per individual cell only. Okay, so you always start with diploid and you end up with haploid. This only occurs in sex organs. Plants, humans, that's where this thing is happening. Okay? So one more time, what is this all about? This is two divisions. Be careful here. The first division, you separate the homologous chromosomes only. So this and this separate. The second division, you're gonna split up the chromatids. So each one of those chromatids here literally splits up and becomes two separate chromosomes. So this is going to be pulled apart from the middle, and each sperm will get half of it. So, any question? Okay, now we talked about the first significance of meiosis. What was the two major things that meiosis gives you and mitosis cannot give you? First, it is the fact that it gives half the number of chromosomes. This is done. Second is variation. The reason why you look different from your parents, the reason why you look different from your siblings, unless you're identical twin, is the fact that we shuffle alleles and we shuffle the chromosomes. So the best thing about meiosis when it comes to variation is that it does two things. First thing, it shuffles up the alleles. So here is somebody who have inherited a pair of chromosomes. One comes from his mom. No, that's the dad actually. One comes from his mom. Okay. Very early in meiosis, one was the first stage of meiosis, was PMAT, right? First stage, it was prophase. Here I'm also going to call this prophase 1. So again, prophase, metaphase, anaphase, telophase. We're going to say prophase 1, prophase 2, meta 1, meta 2, and so on. But again, we're not going to go into details. So assume that this is the very first thing when a cell enters meiosis. We're going to call this prophase 1. Something weird happened. They haven't yet found out why this happens, but the homologous chromosomes of mom and dad pair up like this. This pairing up of homologous chromosomes, we call it, listen to the words, a new word, call it synapses. What does synapses mean? Synapses means the chromosomes line up like this. They become parallel. They are ready now to cross. So they do like this. Okay? They are actually visible. So if you use a microscope, you look at the chromosomes, prophase 1, you'll literally see that the chromosomes are doing like that. We call this the crossover. Mom chromosome, that they do like this. And something very special happens here is that bits and pieces of the chromosome start to be exchanged. So you get an allele here, comes from this side to this side, and an allele comes from this side to that side. Now you tell me, well, how is this important? It's super important because now if some child was born and he received this particular chromosome. So let's say he got this particular chromosome from his parent. He'll have curly hair, blood group O, and blue eyes. Let's assume for a moment that crossing over did not happen. Let's assume for a second that none of this has happened. This time, he would have had what? He would have had the curly hair here. He would have had the blood group O, but he would have looked just like his mom. Everything is the same. But after crossing over, when we crossed over, he got some features from his mom, the curly hair, the blood group O, the blue eyes came from his dad. See how is it important? It basically shuffles, it mixes mom and dad's features together. So you become like more of a combination. Otherwise, if this thing is not happening, you would have looked like a carbon copy from one of your parents. Sometimes it happens, so they look so much like one of their parents, but most of the times you get many, many features of both of your parents. All right, so one more time, what is this crossing over all about? It's basically synapses. Chromosomes line up like this. They literally cross over, they swap bits of pieces of DNA, and you start to, that's a key word, listen to the word, to recombine. We recombine the alleles. All right, so one more time, crossing over, pair of homologous chromosomes, very first stage of meiosis, prophase 1, chromosomes cross, and they make like an X-shaped structure. This has a name. You need to know the name of this X. We call this X chiasmata. Okay? And then, why is this significant? Because if you look at them, that's the only thing you'll see. Then you see bits and pieces of DNA are swapped, and then you will end up with alleles. Mom and dad are recombined. So let me just familiarize ourselves with the new words. Again, the process is crossing over. When we say is prophase 1, what we call it when the chromosomes line up like this, we say that they are doing the synapses. Okay? And this particular structure, we said it's necessary to know, we call it the chiasmata. This 1, 2, 3, 4 chromatids, what do we call it? We say mono, di, tri. What is 4? 4 is tetra. So this is called tetra, the 4 chromatids all together. And this one here, I mean, you can either call it tetra or optional, you may call it bivalent. So if you're referring to 2 chromosomes, we call it the bivalent. If you're referring to all 4 chromatids, you call it the tetrad. Again, you may call them either way. There's no restriction. Both words mean the same thing. Okay. Question? So let's try this. Can you label those chromosomes with the letters?
D is tetrad or bivalent.
Okay, that's good.
A is sister chromatids.
A are sister chromatids.
B is chiasmata.
That's absolutely right.
And C is non-sister chromatids.
Why did I include this? Although it's like common sense, because when you're writing, when you're describing this process, if you just say sister chromatids, they wouldn't understand, or they play dumb. They say, well, what's sister chromatids? Are you talking about this and this? No. You say non-sister chromatids because that's where it happens. It happens between 2 chromosomes and chromatids, but they're not sisters. Those are sisters. Those are not sisters where it's happening. Okay? Now, you get someone's asking what does which one? Crossing over means synapses. No, crossing over is this whole thing. Synapses is when they pair up. The synapses part is chromosome and chromosome paralleling to one another. So here's a sample question. They give you diagrams like the one I have here, and they ask you to show crossing over. The crossover happened here. Okay. So how do you rearrange? How do you recombine those alleles? Let's say I have A, B, D, and E here. A, B, D, and E. Tell me, like, you have done nothing. Yeah, I just copied them because crossing over did not happen here. Where did it happen? It happened right here. So I will repeat that same alleles here as they are. And when it comes to the cross, when it comes to the cuts, I'm going to show that they have been swapped. So I will swap this and this with one another. So this becomes an A, and this becomes a capital A. Okay, everything else stays as it is. Okay, there's another format of this. Here's simpler. If the crossover happened here, you shade, like the outcome is going to look like this, because this is swapped with that. And everything else stays as it is, like this. Okay? Now, there are certain features in humans that they're always passed as a package. Usually, hair color and eye color are always matching. People who come with ginger hair, they tend to have freckles. Many features, they come combined. Very, very, like, you can predict. Someone has maybe blonde hair, usually they tend to have blue eyes. Why is this the case? Because crossing over, although it's random, but it depends on how far the alleles are. I mean, take a look here. A and B are very closely found on the same chromosome. If this crossing over is a random process, think about it. I'm cutting, okay? Where is it more likely to cut? Is it between B and C or it's between A and B? No, I'm far more likely to cut between B and C simply because we have more distance. So one more time, crossing over is random, but you have genes that are distant, then they are more likely to separate. Like C is more likely to cross between two chromosomes. But A and B will always come together as a package. Usually we call such genes that are very closely found on the chromosome, we call them linked genes. So again, what are linked genes? These are genes have to be on the same chromosome and they're always inherited together. Okay? In other words, if genes are more likely to inherit together, they are close. If they are separate, then they are distant or found on totally different chromosomes, which is quite helpful. It helps you to understand how genes are found or where they're found. Are they close? Are they far? Based whether they are inherited together or they are separate. Let me show an example. Here, they gave me this table, and the table says frequency means like chance of crossing over. Think about it. Which two genes are the most distant based on these data? How do you tell? What's the relationship between chance of crossing over and the distance? We said as the chance increases, the distance increases. So the genes that are furthest here on this chromosome are A and B. How far they are? 10. So they gave me here this chromosome, they gave me that chromosome, and they wanted me just to put those genes on the chromosome based on the data. Okay? So A and B are 10 steps away. So I'm going to put 2, 3, 4, 5, 6. Just gonna make it bigger. Maybe disappear. So let me show you here. So this is B. Okay. Am I counting right? 3, 4, 5, 6, 7, 8, 9, 10. Perfect. Okay. Then A and D are 3 steps apart. So I put D 1, 2, 3, or here 1, 2, 3. Both ways are acceptable. I'm just gonna put it that way and then I decide, okay? B and C are 2 steps away. So C is gonna be here, right? Or here. Okay, again, I'm turning these numbers into steps apart from one another. Now, B and D are 7 steps away. B and D are only 7 steps. So which version of D do I take? Do I take this one or I take this one? It must be this, because this one here is 7 steps. This one is wrong, right? I'm just going to erase that D at the end. I'm going to keep this one here. Okay? Erase it that way. But how about C and D? C and D are 9 steps. So do I take this version or this version of C? It says 9. Okay? So this one is wrong. So I'm just going to erase this one. Okay? I'll repeat. So you simply grab those numbers on the top and you put all the alleles or genes on their location. The location is based on how far these genes are from one another. So A and B, I did not have any issue because 10, that's the only option I have. A and D, I put 3 steps away, either way. And then I waited to see other data. B and C are 2 steps apart. B and D are 7 steps. So that made me think that this is the only version of D acceptable. C and D are 9 steps apart. So I erased this one and I kept that. Okay? So just keep this in mind that the greater the distance between genes, the higher the chance of crossing over. Okay? Now, the last bit about meiosis is the second reason of variations. Again, there are 2 major sources of variation. Crossing over was one. Crossing over literally recombines the alleles in a chromosome. The second thing is this. Also meiosis 1, also very early. What comes after prophase? Prophase 1, then comes what? Then metaphase. So metaphase, you get the chromosomes, they line up like this. You see how I line them up? In mitosis, they line up all like this. Here's not the case. The homologous chromosomes face one another. They have to stay one on the right, one on the left, or the other way around. So both options here are acceptable. So this is another version. green on the left, yellow on the right, red here, blue here. Tell me, is this gonna make any difference? I'll tell you yes, big difference. Because the way these chromosomes align, the way they line up themselves in metaphase makes a big difference. And I tell you something that this is random. This is a random process. So the way chromosomes line up in metaphase could result in variation. Because if I am to make these two daughter cells, according to this lineup, you will end up with green and red together because they were both on the same side. So those will stay together. And you will end up with yellow and blue in another cell. So that is one combination. This time, because of the way the chromosomes are lined up here, this time I will end up with yellow and red, green and blue. Now, this is minimum. The only reason why is this not making much difference is because I just have two pairs of chromosomes. But think about 46. Think of the limitless number of outcomes I could have. So this is the second event. This is the second major event that causes variation, which is known as random or independent assortment. What does independent? Independent means here random. Assortment means the way chromosomes move about. So during metaphase, the way the chromosomes line up is random. And when they migrate, when they move to the opposite side of the cell, it will result in new combinations. In crossing over, we said combinations of what? Crossing over, we said combination of alleles. Here, we're talking about entire chromosomes randomly moving in the cells. One more time, what does independent assortment mean? Means random, that's independent, combination of chromosomes during metaphase. I mean, the green and yellow have total freedom to go to either side of the cell. I'm going to show you this one more time. Okay, so let me repeat this. So this is metaphase here. That's the first step, prophase. Metaphase, after crossing over has happened, they line up like this. This chromosome and this chromosome could go this way, right? But the lining up is random. I mean, this and that can swap. And when they divide, when the cell divides, we will end up with what? We end up two cells. These two cells now have one possible combination. Okay? So again, this is my original cell here. This is another one here. And this one and this one have these chromosomes, okay? During metaphase, they could have the chromosome, that's the white, that's the blue, that's one option, and that's the red, and that's the green. Okay, so this is one combination. Okay, you may also have the same cell with totally different combination. I mean, you could have this time the white on one side and the blue on the other side. So here's my cell. That's option 1. That's option 2. So this time I'll put the white on the right, the blue on the left, the green on this side, pink on the other. And when these divide, what's going to happen? Each cell will give a whole new combinations of chromosomes. So here we'll inherit white and pink. Got it? Because they're on the same side. And the other cell will inherit what? The other cell will inherit blue and green. Like this. Same here, if you decide this, if you try this now, you're going to get white and green and blue and red. So that is independent assortment. Finally, if I am now going to meiosis 2, nothing special. Meiosis 2 is literally like mitosis. You take those cells you've made in the first stage and you divide the chromatids. So this chromosome went here. One goes to the right, one goes to the left. Same idea here. Again, I'm done with all these special events. Everything that happens in meiosis 2 is like mitosis. I split the chromatids apart, and then I end up with what? I end up with gametes. Each inherits one copy of these chromosomes. Just take a look at these gametes we made. Notice, none of them is same as the other. Each one is different. So like this one has all blue, this has a patch after crossing over. Same idea. This one is all red, this one has a patch of blue. So this tells you that all of the genetic variation happens in meiosis 1. Meiosis 2 is just process to produce those 4 gametes. Okay? So let me recap on what we did here. We did interphase. The major thing about interphase was copying of the DNA. We duplicated DNA, S phase. We did meiosis 1. The most important thing is the crossing over and the independent assortment. During crossing over, what was the key word here we use when we talk about crossing over? We say recombination of alleles. And during the meiosis 2, we form the gametes. That's the time when we make those 4 sperms or gametes. And that's the time where we separate the chromatids. That's the time when we end up with individual chromosomes looking like this. Just compare this one. Just compare the cell we made at the end with the cell that I've started with. Literally, it's half the amount of DNA. Okay, this goes with crossing over. Let me do quick comparison between meiosis 1 and 2. So, synapses, do you guys still remember? Just tell me in the chat, tell me 1 or 2, okay? Was this in meiosis 1 or 2? What do you think? This word means chromosomes parallel, so that's 1. Crossing over, 1 or 2? Okay. Independent, too many ones, right? Is this 1 or 2? Still 1, okay? Again, all the events, all the special things happen in 1. 1 or 2? 2. That's when we separate chromatids. Separate homologous chromosomes. When do we do this? When do we only separate the homologous chromosomes? What is this doing here? You still remember this from last time? Cytokinesis is splitting the cytoplasm. That's when the cytoplasm, the cell splits between the daughter cell. This happens in both. I'm going to put it right here because we divide the cell initially here into 2 cells, and then we divide this cell into 4 gametes. So cytokinesis, division of the cytoplasm, happens in both cases. All right. I want you here to put check marks between meiosis 1 and 2. It says pairing of homologous chromosomes. Is this 1 or 2? What do you think? Do you know another word for this? Do you know another word for pairing of homologous chromosomes? We have one word to describe this whole thing. Call that. So I'm going to put this one here. Each chromosome consists of pair of chromatids during prophase. Think about it. Chromosomes are like this. Chromosomes are attached like chromatids in the middle. Where is this? This is a cell. This is a cell in prophase 1, okay? This is a cell in prophase 2. When do we separate the chromatids? We separate chromatids in anaphase 2. So just be careful. So either one is still attached, okay? Now, crossing over occurs and chiasmata. You know this one here. Independent assortment, 102. Okay? Now, if you're wondering what's the difference between mitosis and meiosis, let's make a quick comparison. Which one was two divisions? You know this one. Where am I supposed to put DNA replication? Put T or E, please. T or E, based on what you think. TE. This goes for both, right? Don't we have S phase? Don't we have interphase for both? Which one results in genetic variation? I flipped it, sorry. This goes here. Okay? How about this? Here or there? Goes here. Two diploid cells are made. Two identical diploid cells, it goes here. Separate chromatids. We do in both, right? Here it only happens in meiosis number 2. Okay? Pairing up of homologous chromosomes, this is here in M1, meiosis 1. And separation of homologous chromosomes is also in M1. Here I have a summary of this whole thing. So let's try. During meiosis, chromosomes pair up. So which word is the most appropriate here? Which stage I mean here? This is it was the very first thing, right? So I'm gonna go with prophase 1, okay? The pairing up chromosome is known as what? What was that word for chromosomes pairing up? This was here. Fragments of non-sister chromatids are exchanged. This process is known. So this is crossing over, which leads to, what does that lead to? It leads to the recombination of alleles. Now, what do I call this structure that is visible and you see at this stage? So something like this is visible. What do we call that? This is known as the matter. Now, during homologous lineup randomly, during what? M or N phase? Each one of the chromosomes has freedom to move to either pole of the cell. During anaphase 2, that's very important, that anaphase 2 is when sister chromatids separate. This results in the formation of what? I'm gonna put haploid. And I say here, that's important. Do I go for the word genes or alleles? Must be this one, right? Why allele? Because me, you, everyone, every human, every Homo sapien has the same genes. But how are they different? We are different by means of our alleles only.